Welcome back to SCH 214, Intermediary Metabolism 2 and Biomembranes. Today we are continuing with biomembrane structure and function. The last time we did look at biomembrane composition. So we're looking at the structure and function today. And this is going to be the last part one lecture because I understand from 18th July, you start to write your exams. So we'll have one more lecture next week, and then you'll get ready to take your exams. Right. Right from the beginning, we want to look at the fact that biomembranes have a structure of what is referred to as a fluid mosaic model. Now, first of all, what's a mosaic? So a mosaic is an artwork which is made up of different components. So like this mosaic, which is on the right-hand side, where some stones and different objects have been used to do an artwork of some kind of a design. So similarly, you have cell membranes, biomembranes made up of different components that come together. We did see the structure of biomembranes the last time, where we saw it's a lipid bilayer and you have some proteins in it and some other components of complexes of lipids with proteins, complexes of carbohydrates with proteins. You saw cholesterol and other things together. So all of these come together to form what is being referred to as a mosaic structure. Now we say it's fluid because that structure is flexible, it's not rigid. So that is why we refer to it as a fluid mosaic model. Of course, the main component in there is the phospholipid bilayer, in which everything else is embedded. And so, I mean, that's what we do refer to as the fluid mosaic model. We have seen it over and over. We just didn't use the term fluid mosaic model until today. But we can go on and look at what we are talking about much more. Again, just like we have seen before, we did see that the biomembrane, I mean, say the cell membrane is made up of a phospholipid bilayer. So what you see there, the phospholipid bilayer, where in this case, the representation here is showing that, above it, you have outside of the cell. So that is the extracellular space. And the below this structure is the cytoplasm. I mean, this is just to depict that you have some structures inside of the cell in the cytoplasm and some structures outside of the cell in the extracellular space. And then you can notice that in this lipid bilayer, you have some different proteins that are embedded. So you have some peripheral proteins. So these are proteins that do not transcend the whole thickness of the cell membrane. So these green structures are peripheral proteins. And you also see that on these peripheral proteins, you can have, for instance, some carbohydrates, like some oligosaccharide. So this structure will be acting as a receptor for cell recognition, cell communication. You also have some proteins that are, transmembrane proteins. They transcend the whole thickness of the cell membrane. So you have that protein being shown where you have some alpha helix, which is embedded within the cell membrane thickness. And then you have some loop outside of it, both are the extracellular space and the intracellular space. You also notice there's a transmembrane protein, the red ones. These transcend the whole thickness of the cell membrane. And here too, you see that you have some carbohydrates that are attached there. So that will be a glycolipid there. That a glycoprotein. That's a glycoprotein, or also acting as a receptor. You can have even some carbohydrates, like some oligosaccharides, which is attached directly to, say, the phospholipids that are components of the lipid bilayer, and that's a glycolipid, also acting as a receptor. Then, of course, we did see so then you do have also other components like sphingolipids. So apart from the phospholipids, you can also have some sphingolipids in there. So all of these, you can see, these are different components which are coming together. Just like we saw with that mosaic artwork, we see that these are different components that come together to form this whole structure. And the structure is not rigid, it is fluid. And that is the reason why we refer to the biomembrane as having a fluid mosaic structure. Now, if you look at that fluid mosaic, you will realize that we did talk much about the structure and functions of lipids already, even from last semester when we were looking at intermediary metabolism 1. We did look at the structure of lipids, looking at the fatty acids, and then different aspects of it, having the carboxylic acid group as the hydrophilic part of the molecule, and having the carbohydrate tail, which is the part of the molecule which is hydrophobic. You have that hydrocarbon chain there. We did look at all those lipids, sphingolipids, phosphoglycolipids, all of that. We did look at it extensively, and even the last lecture we did look at the structure of sterols, which that of the cholesterol. We did see that cholesterol is what you find in animal cells, and you have phytosterols in plant cells. So you can see we did cover everything about the lipids that are found in the structure of the biomembranes. So what is left for us to look at here mainly is the proteins, that are embedded in the lipid bilayer. So today we'll focus more on the types of the proteins that are embedded in the biomembrane. Now you do have here different types of these proteins in this biomembrane. We have what we refer to as the integral or the transmembrane. Also, we refer to them as intrinsic proteins. So if you hear these three terms, they are referring to the same thing: integral or transmembrane or intrinsic. Then we have the peripheral or extrinsic. So these two, they are also, if you hear either peripheral or extrinsic, it's referring to the same thing. We have the lipid anchored proteins and we have associated proteins. So we're going to take a look at each of these types of proteins in the biomembranes. Now, what you do see as the peripheral proteins are these proteins that do not transcend the whole thickness of the biomembrane. So for instance, you do see this peripheral protein, which has, say, halfway, being embedded in the membrane. It hasn't transcended the full thickness of that membrane. So that is a peripheral protein. Here's another peripheral protein, which has not even embedded itself into the lipid bilayer, but just at the surface. So that's another type. That's another type of peripheral protein. You have so this is an integral protein. So again, if you hear the integral protein, is the same as a transmembrane protein or intrinsic protein. So you see that has transcended the whole thickness of the membrane. Then you see another one which is associated. So this is the protein which is not attached directly on the phospholipid bilayer, but it is attached to an integral protein. That's what is referred to as an associated protein. It's also peripheral, but it's because it doesn't transcend the biomembrane, but it is attached to another protein. That is why, which is integral. That's why it's being classified separately as associated protein. Then you have the last one here, which is anchored. So this is a protein which is anchored to a lipid. So a lipid, there's have this short lipid which is embedded within the lipid bilayer, and then attached to it is a protein. So that protein is anchored on that lipid in the biomembrane. So I mean those different types of proteins that we were looking at on the previous slide. This is a pictorial depiction of all those types of proteins. Now, taking a closer look at it, if you have a biomembrane where you have outside of the cell, which is above, this model, which is to show that, above it is the extracellular space, and below it is the cytoplasm or the intracellular space. Then you can have, a protein that is embedded on a lipid. That is attached or anchored to a lipid. Now, what you do see here is that we're showing the bonds. So, for instance, if you have a lipid on which the protein is anchored, and that lipid, of course, is having a carboxylic acid group. Let's say it's a fatty acid, and it's having a carboxylic acid group that interacts with the amine group of the protein. So see, the N-terminus of the protein forms a peptide-like bond with the fatty acid. So you see that the C-terminus is at the end of that protein. You can also, take a look at the integral proteins. We will see different types of even these integral proteins. But this is just to depict one of them, where you have a protein which is folded, several times. So you have these several portions of the protein which are embedded in the lipid bilayer. The protein is folded severally, and you have such domains, these alpha domains, which are embedded in the lipid bilayer. And you see the N-terminus is at the extracellular space, and the C-terminus is in the intracellular space. But this is just one type of integral proteins. We'll zoom in on these integral proteins and take a look at the different types that you can actually have. Now you see the types of the membrane proteins here, where we have type 1 up to type 6 of these membrane proteins. Now, type 1 is a membrane protein that is having just one loop. Let's say that is one alpha helix, which is embedded in the lipid bilayer. And you have the N-terminus at the extracellular space. Take note that above the structure is outside of the cell, and below the structure is inside of the cell, which is to depict that you have an extracellular space above, outside the cell, and an intracellular space, which is the cytoplasm. Now you can have transmembrane proteins that have the C terminus in the intracellular space in the cytoplasm. Then you have the N terminus at the outside of the cell. And vice versa, then you have type 2. So type 2 will have opposite of type 1, where you have the N terminus in the intracellular space and have the C terminus at the extracellular space. You do also have type 3, where, as you can see, you are having multiple loops, multiple alpha helices, and these alpha helices are embedded in the lipid bilayer. But in this type 3, you can either have the C terminus at the outside or at the inside of the cell. So you have this interchange. You can have any of them. The main difference for the classification here, the main distinction of this classification is that you are having multiple loops that are embedded in the biomembrane. Multiple alpha helix domains which are embedded in the lipid bilayer. And type 4, in type 4, you have these. So if you pay attention to type 4 and compare to type 3, you realize that type 4, these are single proteins that are having alpha helices embedded in a form of a cluster inside the biomembrane. And by type 3, it is only one protein that has folded multiple times all the way to the end. But if you take type 4, you see that each of the proteins has a C terminus and an N terminus, and they are clustered together in that form. Then you also have, so this is type 5, which we have seen the anchored protein. That's just anchored. In this case, this anchoring is on a lipid. Sometimes you can have such an anchoring even on a sterol. So you have type 5 having such, structure where you just have a lipid that is embedded in the biomembrane and a protein that is anchored on it. If you compare that to type 6, you realize that type 6 is both anchored and at the same time it is transmembrane. You have this alpha helix which is embedded in the biomembrane, and you have say the C terminus inside of the cell, whilst you have the N terminus that will be forming some peptide-like bond with the lipid there. And so you have all of these from type 1 to type 6 membrane proteins. So you want to take note of that as the types of proteins which you can have in biomembranes. Now we can move on to take a look at the functions of biomembranes. And a major function of biomembranes is transport, material transport into and outside of the cell. And this is responsible for what we refer to as a nutrient uptake. You do remember that, for instance, in the small intestine, you do have these structures, finger-like structures, we refer to as villi, that you have absorption of nutrients from food through these villi into the bloodstream. Now, that barrier of the small intestine with those villi, of course, they are made up of cells. And you have, some membrane structure through which you have, the uptake of these nutrients into the bloodstream. Now, but even apart from that, if you take a cell, specifically just a cell through which you have some, substances that will be absorbed into the cell and then outside of the cell, you do know the cell is the basic unit of life. So if you have nutrients that get into the bloodstream, into the system, those nutrients eventually will have to be absorbed into the cells for cellular metabolism to take place. Now, you can have, primary molecules that easily move across cell membrane, being, say, water, carbon dioxide. Carbon monoxide can also go across the cell membrane. You have oxygen. Mainly the compounds which move across cell membranes for metabolic processes. Yes, you have oxygen for cellular respiration. Water is a major component that is always moving across the cell membrane. Carbon dioxide, a lot of times, that's a byproduct which is produced from cellular metabolism. So that also moves across the cell membrane. Now, there can be some larger molecules as well, like biomolecules, like some carbohydrates, because of course carbohydrates will have to move across the cell membrane, get into the cell before they undergo some metabolic processes, breaking them down there, or maybe synthetic processes, as we saw with metabolism of the various biomembranes. Now, what you see on the right-hand side is that you can have, say, maybe fat-soluble molecules that can diffuse across the cell membrane easily. And that is because you have the cell membranes being a phospholipid bilayer. And that phospholipid bilayer is lipophilic. So if it's lipophilic, it means that, lipids can easily diffuse across it. Small molecules of lipids will easily diffuse across it. How about water molecules, for instance? So water molecules will have to be transported through some special protein channels because water and the lipid will not mix. Just how you have seen the fat-soluble diffuse easily, water will not be able to do that. And so you have some channel proteins that are involved in the in the movement of, say, water molecules across it. We'll take a look at we'll zoom in on, say, mechanism of movement of water molecules across, say, these types of channels much more closely. Now, you do have what we refer to as channel pores. So these are proteins, transmembrane proteins, responsible for the transport of ions across the cell. So that's a channel, an ion channel or pore that has some sort of a pore within the transmembrane protein through which you can have substances moving from, say, above it. Again, here is an extracellular space, and then below it, an intracellular space where you have some ions passing through from the outside into the inside of the cell. So you do have these types of channels. Then we have what we refer to as transporters, protein transporters. So, if you take a look, you realize that this channel, this ion channel, or these ion pores do not have what we refer to as the mechanism we're seeing here is some kind of a gating. I will actually come to it much more closely for you to see the difference between these pores and transporters, even though sometimes they are used interchangeably. Here you can see that you just have a straight pore through there, that you have a substance that fits into the size of that pore and it can pass through it. So you have substances passing through these pores depending on the size of these substances, or that is the ions. If you come to the transporter proteins, you realize that the mechanism of transport here is much more complex than what we have just seen with these pores. You realize that you have the transporter protein opening at the extracellular space while closing at the intracellular space to allow for some specific ion to get into it, some specific substance, which is a bigger substance. So usually you have these transporter proteins transporting much more bigger substances than the channel pores. And you see once that substance gets into the transporter protein through that opening, you see that the transporter protein begins to close to the outside, whilst the inside opens. So you see in the last end, you see that the transporter protein closes at the extracellular space while opening at the intracellular space to allow for that substance to move cell. It's a much more complex mechanism than this the ion channel transportation, where the substance just passes through it directly, depending on the size. It can get into the pore or not. But here you have, although it also depends on the size, you do have a certain system where you have one side, that is the extracellular space, opens while the intracellular space closes. It's not only this mechanism. As we'll come to see, there are different types of these transporter proteins. All right, so that is one aspect of transportation across these proteins. Now, another thing we need to understand is that you have these transporter proteins, so say the pores, the ion channel pores. You do have different forms of transportation of substances in such a way that, we have what we refer to as protein ports. We have three types of them. That is uniport, antiport. So, sometimes you also see in biochemistry books as uniporter, antiporter, symporter. It's referring to the same thing. Sometimes the term can be used in such a way that the uniporter, antiporter, and symporter is referring to the proteins that are embedded in the cell membrane, whilst the term uniport, antiport, symport is referring to the process of transport across these proteins. So that a uniport refers to the movement of substances across the transmembrane protein in one direction. So you see the movement of particular substance only in one direction, say from the extracellular space into the intracellular space. So that's a uniport. It only allows for one direction of movement. Now you have an antiport that allows for movement of two or more substances in opposite directions. So as you can see, you have these substances depicted with the green triangles that are moving from one side of the cell membrane to the other side of the cell membrane, while another, which is like a hexagon, is moving from the opposite side, to the other side of the cell membrane. So that's an antiport. A symport will have two or more substances which are moving in the same direction. So you have these violet and brown stars depicting different substances, but these substances are moving from the same direction, from the same location through the same direction, through that membrane protein. That is a symport. So you have these types of protein ports as well for transportation in different directions. Then we have also what we refer to as ionophores. And these ionophores can either be electroneutral or electrogenic. Now, what do these two terms mean? Now, electroneutral means that these ionophores allow for the transport of charges across them, say equal charges in opposite directions, so that in the end, there is no net change at the opposite sides of the membrane. So for instance, if you have sodium ions as Na plus as X moving up, and you have potassium ion, which is K plus. So both of these ions, these examples of ions are having +1 charges, and they are moving in opposite directions. So in the end, you don't have any net charge across the membranes. Now, an electrogenic ionophore will have transport of ions in one direction, such that in the end there is a net charge at one side, or say on both sides, because one side is going to be, say, positively charged, whilst the other side is going to be negatively charged, because you have movement of charges from one side to the other, which is not compensated for with that same ionophore. It can be compensated for by some other system of transport, but this particular system that is transporting the ions is not compensating for it. So you do see that opposite, that for electroneutral there is no net change in charge, but for the electrogenic you have a net change in charge. I mean, say for instance, maybe if you take say an ion channel that is say transporting let's say so potassium and hydrogen ions in one direction. Both of them are having +1 charges, and they are moving in one direction. So obviously the direction to which they are moving will be having a positive charge, whilst the location from which they are moving from will be carrying a negative charge after those ions have been transported. So these are two types of ionophores that we have: electroneutral and electrogenic. And the structure of the ion channels are such that you have these ion channels being made up of some protein domains. I mean, on the left-hand side, you have these protein domains, which are depicted, with different colors for us to see that, like you have a cluster of proteins. You see what we did see with those proteins, which can be in a cluster, embedded in the biomembrane. It's a similar thing. So you have these proteins embedded in the biomembrane. So it forms something like a funnel through which these ions can pass through. Now, if you look at it much more closely, you have these structures made up of protein domains which are labeled 1. So these are different domains of proteins which come together and form something like a funnel, through which you have these ions passing through. And then you have what we refer to as the channel opening. That is the space through which you can have the ions passing through. And now, number 3, you see what is being referred to as the ion selectivity filter. Now, the ion selectivity filter is some amino acid residues. I mean, you do remember we've dealt with proteins, and so much you understand things like maybe active center of enzymes, where you have specific proteins that can, you have specific amino acids that can interact with substrates. I mean, in a similar fashion, you have some amino acid residues present in a specific portion of this ion channel, and these specific amino acids can have some interaction with substances that are getting into the ion channel. So, depending on the nature of the substance, those substances can be can either be attracted. Maybe depending on size, they can move through it, and at the same time, depending on chemical nature, there can be a repulsion between a substance and this selectivity filter. So that's it's not every substance which is allowed through this selectivity filter. Then you do have number 5, what is referred to as a door, or that is a channel door, or it's a phosphorylation site. I mean, we did see how the process of phosphorylation acts as a form of activation of biomolecules, to get into some specific biochemical reactions. And here you do have at some specific ends of these proteins, we did see that you can have, say, N terminus, C terminus of these proteins, which are either at the extracellular space or the intracellular space. Now, these N terminus, C terminus can act as some specific sites that interact with some energy molecules like ATP, and the phosphorylation of the protein on those sites, activate the ion channel. So you can have the ion channel opening or closing depending on that process of phosphorylation to allow or not to allow substances to pass through these ion channels. Then you have, of course, number 6 is just showing that that's a cell membrane, across which you have, this ion channel embedded. Now, this is just to show you some examples of the ion channels we're referring to, where you have, again, above, it's an extracellular space. Below is the intracellular space, where you can have what is referred to as the potassium channel, which is, responsible for transportation of potassium ions from inside of the cell to the outside of the cell. You can have sodium channels that are transporting in the opposite direction. Usually you have potassium-sodium channels working hand in hand. And you also do have, if you just apply what we just looked at, it means that this channel, potassium channel and sodium channel, these are unipots. They are transporting these ions in only one direction, whilst you have this cation channel, sodium-potassium channel, which is, we can say sodium-potassium pump, for instance, is transporting, say, potassium ions from the intracellular space to the extracellular space, whilst transporting sodium ions from the extracellular space to the intracellular space. So that's an antipod. That's an antipod. And you can even also then say that this channel, if you think of it as in terms of an ionophore, then this channel is electroneutral. Equal charges moving in opposite directions. So in the end, this transport is not going to result into a net change in charge across the membranes. And you have the calcium channel also transporting calcium ions in one direction. And so that too is a uniporter. The chloride ions, the same, a uniporter transporting in one direction. All right. Now we can take a much closer look at, the transport of water molecules by these transport proteins we refer to as aquaporins. And you see here, these water molecules that get into the transport protein. And you can see, what we were referring to much earlier, some specific amino acids, So you see there's some amino acid which is at position 180. You have another one which is the N-terminus is at position, so that the amino acid is at position 76. Another one here. So I mean, you do remember that we refer to say the proteins, the amino acids that are in them. We refer to, say, if you refer to the primary structure, you can actually count the amino acids. They are the sequence of these amino acids. You can relate to the particular position of these amino acids. And you realize that at these specific amino acids, at the 180th position, 192nd position, 76th position, these act, with having some positive charges that have some form of interaction with a water molecule, giving it a specific orientation for it to be able to pass through the channel. And you can see there's some constriction there, which is referred to as the narrowest point, which results into either, if a molecule is much bigger than, say, water molecules and is not allowed to pass through here, of course there's going to be some repulsion, and that molecule is bounced back. It does not pass through these aquaporins. And so you have what is being referred to as some electrostatic repulsion, but this kind of repulsion is particularly for orientation of the water molecules, because the water molecules some hydrogen and oxygen that is forming a dipole structure. And so gradually you have such a movement with orientation of these charges at the various amino acids that are controlling the movement of the water molecule through these aquaporins all the way until these water molecules pass across from one side to the other. Now we do have what we refer to as ion channel gating. The examples we saw much earlier with some transport proteins, we did see that you could have one side of the channel closed while the other opens. And here we do have what we refer to as a single gating. So there are some ion channels with single gates, and what it means is that you have, say, for instance, only, say, the extracellular space that is having these membrane proteins closing. And when there is supposed to be a transport of a substance, it is only that gate that opens for the substance to get through the channel to the other side of the membrane. There's only one gate. That's a single gate. Now you also have what is referred to as the alternate gates, where you have the transmembrane protein having two gates. I mean, at each side, the extracellular side and the intracellular side. And you see that when you have, say, one side of the gate closed, you have the other opened. So you can have a transport of substances that are coming from the other side, maybe from the top, that gets into the membrane gets into the transport proteins embedded in the membrane to the other side. So you see, when you have the other side opens, if you take the up as the outside of the cell, the in as the outside of the cell, then of course, when you have the extracellular space gate opened, then you have the intracellular space opened. And when you have the extracellular space gate opened, you have the intracellular space closed. So you have what is being referred to as alternate gate opening. So you have such alternate gate opening there. So these are alternate gates. Now we also have what we refer to as ligand-gated ion channels. So these are channels that are controlled by biomolecules that are acting as signaling molecules. I mean, usually you find some proteins, some neurotransmitters like acetylcholine esterase that bind to the surface of some specific receptors, and it will trigger the opening of some channels for some ions, which will then result into transmission of some electrical signal in nerve cells. And so you have these ligands, being referred to as chemical messengers. Like if you have this acetylcholine, which is binding on this protein. So you see, first of all, the transmembrane protein is closed. There's no movement of any ion through it. Then you have this brown spherical structure, which is depicting a chemical messenger. For example, a neurotransmitter, as I said earlier, binding on the receptor of that transmembrane proteins. It then triggers the opening of that that channel to allow for sodium ions to pass through it, across it. Yeah, so you do find this type of channel gates, the ligand gates, with say potassium, sodium, calcium, and chloride ion, channels, proteins that transport these ions. Of course, this is to say that you have these proteins, the transmembrane proteins, actually responding to that binding of the chemical messenger or the ligand to it. That's a form of cell communication or cell recognition. But cell recognition will be to recognize maybe some specific substance that is entering into the cell, particularly if there is some invasion by some bacteria, virus. You have some of these receptors on cell surfaces. This particular one is for these ions. This response is for these ions, and this is for like nervous system responses. Then you have the voltage gated. Now in the voltage gated, you can see that there is no chemical messenger which is binding onto the membrane protein. So you have this is a membrane protein, a transmembrane protein, which is having a positive side, a positive charge on, say, the extracellular space and a negative charge in the intracellular space. You can see it's having a gate in the intracellular space, like a single gate there. And I mean, in response to, say, the movement of sodium ions across. So this time around, ions, when they move across the membrane through these channels, they then result into difference in charges across the membrane, like what we saw with the electrogenic. So these are electrogenic, channels. That you see, if you have a sodium ion that is moving across it, then of course, on the other side where the sodium ion goes, there will be an increase in positive charges, which will create a negative charge on the other side of the membrane, which is why it's being referred to as voltage-gated ion channels. So these ion channels open as a response to the difference in the voltage or the charges that results due to the movement of these ions across these membranes. Right. Then we have some channels that respond to say what we refer to as stretch. to say, I mean, it's a physical difference in conformation of these proteins will result into some specific biochemical response. So you see, you can have light, temperature, and then some mechanical deformation, which can result into some specific biochemical responses. So, for instance, you have these say rod-like cells, which you find in the retina of the eye, which respond to say light. So when light, because we see by light entering into our eyes. So when light falls onto these proteins, these in the rod-like cells, this orange structure is what is depicting, say, these particular proteins. Rhodopsin, the protein is rhodopsin. When light, strikes it, it opens, and when it opens, it allows for sodium ions to pass through it. And once these sodium ions pass through it, you have differences in electrical charge, which is resulting across this cell membrane, which then results into electrical signals across the nerve cells, which will then be passed through the visceral nerves to the brain. And so we recognize things by sight. So that is a mechanism of sight, which is at the cellular level. I think I have an animation of this that I'll send to you, so you can see what it is. Here is just an image. So when I send you the animation, you'll see it much more clearly. Temperature and say mechanical deformation. So this is for temperature, where you have receptors. So you see, you have 4 different proteins which are clustered together, forming that channel there in the membrane. These are what we refer to as some vanilloid receptors, TRPV1, some vanilloid receptors. Sometimes, okay, referred to as maybe a transient receptor protein. And these receptors recognize, say, temperature, heat. They are the same receptors that also recognize, say, the heat from, say, chili pepper. Pepper, when you take in pepper, you feel the heat in it. I mean, you have these receptors that are responsible for recognizing such sensation of heat also from pepper, and a particular compound which is responsible for that heat in pepper is capsaicin. Now, what you see is that you have these vanilloid receptors. When they recognize, say, such heat, they open up, allowing for some hydrogen ions. There can also be movement of sodium ions, calcium ions, which then results into change in charge across the cell membrane, which will result into the movement of, say, some electrical signal along the nerve cells into the central nervous system. So you see that movement into the central nervous system, and the burning sensation, or maybe even some kind of an itching or stinging on the skin. For instance, if something stings you, an insect stings you, or anything of that sort, it is by this same system that you have that a signal going to the brain, and so that the brain then translates that, and you feel what it is as either some heat on your tongue, or maybe it's heat in the atmosphere that you are feeling on your body. You have such a mechanism being referred to as also as a stretch activated. Previously we did look at a stretch activation by light. This is by heat. We can look at also by mechanical. So I think this should be the last one. After this, we can end it. Where you have sound. So how do we hear? You have sound which gets into the external, outer ear through that channel, the ear canal. You have the sound going through to hit the eardrum or the tympanal membrane. Now, when the sound hits that membrane, it causes some 3 small bones in the middle ear. The malleus, the incus, and the stapes. These are some 3 small bones in the middle ear, so that when you have sound hitting the eardrum, it causes vibration of the malleus. And that vibration from the malleus is transferred through the incus, that's the middle bone, to that the third bone there, the stapes, which presses against the inner ear. What is being referred to as the cord part, the cochlea, but inside this whole structure of the inner ear is that auditory fluid. And because of the pressure that this bone, the stapes, will have on the inner ear, it will cause vibration of the fluid inside the inner ear. And such a vibration in the inner ear will then cause the movement of some hair-like structures inside these cochlea. Cilia, we do call it a cilia, and the movement of such hair-like structures then sends some signal for the movement of, you have some neurotransmitters that move across the cell membranes, and you have this electrical signal that moves along the auditory nerve, and the auditory nerve then to the nervous system, that is the brain, so that whatever it is you are hearing is then translated by the brain for you to have an understanding of the sound that is coming to you. I think these two, I have some animation which I'll look for the link, and then I'll send it to you so that you can look at the mechanism of hearing by this. So you see, if you put your mind to it, you realize that, for instance, deafness can occur in by different ways. It is either you have eardrum damage. If the eardrum is damaged, then of course, if sound hits it, there will not be so much of vibration, because it will be like the air will just, the sound which is coming from outside will just be passing through the holes inside these eardrums, which can actually even be piercing and causing pain. So such persons will even have to be closing their ears. Or you have some deformation with the middle ear, where you have these 3 bones being defective and not carrying out their function properly. Or you have a problem with the inner ear, either with a fluid which is inside. If you have the inner ear damaged and there's no fluid in there, then of course, even if there is sound that hits against this, the inner ear, there will not be any fluid that will move to cause some form of a ripple effect, which will result into the movement of the hair-like structures there to cause some electrical signal transduction, which will go across the auditory nerve. Or you can have the auditory nerve itself, the damage of it also, meaning that even if all these parts of the ear work well, but the auditory nerve is defective and cannot carry the signal to the brain, then there will also be impairment to hearing. So with this, you can actually relate to the different forms of deafness that can occur. All right, so I mean, this will be where we'll end it, and I wish you all the best till we meet next time. Bye for now.